Wow, thank you everybody. My presentation is going to be a little bit different, as you may imagine. We do have some data, but what I want to do today is to really tell you the progress we've made over the last 14 months in being part of the Oxford-Harrington partnership. Yesterday, I think we all took inspiration from Lord Cameron, Baroness Blackwood, Sir John. I was particularly impressed with the eloquence of Julia, Theodore, and actually this morning Mel, if she's still here, so thank you for that. I was actually reassured and somewhat excited by the CEO of the MHRA this morning, which is probably the first time I've said that in 25 plus years, but thank you, Dame June. And actually, I have the privilege to lead this company. The work behind it spans many, many years, lifetimes of work of some of the leading academic institutions in the UK. And actually, coupled on my own personal experience of one of my daughters who had a very difficult time when she was born 20 years ago now, who was magically revived by the JR and ultimately fixed at Gosh, and who is now also a third-year clinical undergraduate at Birmingham University studying medicine. So I feel I've seen life from both sides, and I can perhaps put a unique perspective on these things. I have built a respiratory company before. We'll talk about that maybe another day. But today's focus is AlveoGene and what we're doing in transforming rare disease using inhaled gene therapy. Please work. Right, down, not up. Okay, there we go. So, who are we, all right? We are a unique lung-targeted inhaled gene therapy. That's really key. We target the young. And what our aims are to do is to transform the standard of care, or indeed, in certain cases, provide therapeutic solutions where currently there are none. Our platform we call Ingenuity. That's an acronym for inhaled gene therapy. And please don't worry about this, but this is a third-generation self-inactivated lentivirus, which we use in vivo and directly, that we have shown that uniquely combines efficacy, safety, durability, which is absolutely key, all together with a potential single-dose delivery. The preclinical data we have is utterly compelling. I'll highlight a couple of those in our surfactant protein deficiencies and our alpha-1 programs. And this indeed supports onward clinical development, which, as Diana said, now we're at that accelerated stage, is our next challenge. The pipeline, which I'll show you as we believe we have lasting billion-dollar commercial potential. And again, based on the work of the previous academics, the technology is largely de-risked. Prior to AlveoGene's formation, the guys managed to do a very large deal with Boehringer Ingelheim using the very same platform, the very same CMC in cystic fibrosis. And that, to us and to any other investor, I speak as an engineer in the past, but CMC in this space, as we've heard over the last day and a half, is absolutely critical. We're on a Series A march now. We're attempting to raise about 50 million to take 2 or 3 programs forward. Our first programs are in surfactant protein deficiency B and ABCA3 deficiency, which I'll highlight. We probably have the best candidate, which will ultimately be the preferred product for alpha-1 antitrypsin deficiency in the lung. I'll show a little bit of data on that. And because of the unique features of the opportunity of the vector in its own right, those new features of the vector in its own right, we do actually have capability in some interesting fibrotic diseases, particularly IPF, because of the packing capacity that our vector allows. We've talked a lot about altruism, I think, over the last day and a half. We've had some suggestions on not-for-profit organizations as well this morning from Chaz. And maybe there's some other opportunities. But I have to say, it's the cold, hard reality that when you're taking investors' money, they will also expect a financial return over and above their impact. And I think we can be confident that we can hit both things. So this is our pipeline on our Series A funds. We are relentlessly focused on actually achieving clinical success within 3 years from today. We have an extremely good chance of achieving that. Actually, 2 potential marketing authorizations in our surfactant programs, led by Professor Deborah Gill, who's kindly in the audience today of Oxford University. We have been accelerated, as I said. We are the first, we're the poster child of this program, and I think the team have deployed the funds very well, very capital efficiently, and now we're on to that next stage of our journey. Supporting that, we've got equally impressive work ongoing in the alpha-1 space. That's a little bit more crowded, as I'll explain a little bit later. And we've heard a number of different ways of approaching that disease. I think yesterday we heard a CRISPR presentation of a knockout in certain things. In alpha-1, you've got to knock things back in, which is a little bit more complicated. But in the round, when we come to lung disease, which is the most prevalent form of alpha-1, 80% of patients have lung disease, as opposed to 20% in liver patients. I do believe we'll ultimately get there. But our goal over the next 3 years is to actually complete a phase 1 human trial. In 4, we have, this is the work up at Edinburgh. This is in an earlier stage, but again, in the combination of maybe multiple genes and microRNAs, we can end up with a very highly differentiated candidate and one that should work. So what is Ingenuity? Well, it's a fusion of 2 viruses. The Sendai virus is highly tropic to the lungs because of its FHN surface proteins. Mouse flu, if you want it in a different way. And of course, with most viruses like this, it goes in well, but its effect wanes very quickly. So it's quite good for Nature papers, as Eric tells me, but it's rubbish for durability and longevity. The lentivirus, however, as we will all know, integrates very well. However, the surface proteins of the lentivirus are not really tropic to lung epithelial tissue. So the moment of genius, which was had a few years ago, is why don't we strip off the surface proteins of the Sendai virus, strip off the proteins of the lentivirus, and replace them with the surface proteins of the Sendai virus, and you will end up with this RCIV FHN lentivirus, which is brilliant. It is by far and away the most efficient way to deliver genetic material to the lungs across the full lung, across all type of lung cells, as you can see from the staining top to bottom, from the ciliated cells down to the type 2 cells at the bottom, sometimes in the alveoli, where we're targeting. We have 2 forms of delivery. I'll speak about neonate installation. We had a lot of neonate stuff. There was some, the genetic newborn screening program. How can we get to this point? Today, if we did that genetic screen, it would come back in a couple of weeks. We could deliver this therapy to this child and they would live. Right now, they die. It is that binary in terms of what we can do. In the AAT space, however, also speaking as an engineer, I'm pleased to report that our formulation of a nebulization solution is very strong. It survives the nebulization process. It remains very potent. And this is how we will do it for alpha-1 antitrypsin deficiency using a standard breath-actuated nebulizer. What are the advantages of Ingenuity? I think top to bottom, it's very efficient in its conduction because of its parents from the FHN surface proteins. And on the top right, if you can just see on the GFP photograph here, a whole gamut of species have been done, including ex vivo maintained human lungs. The duration of expression is phenomenal, best in class, without question. You can see here 2 years of life of continued expression of therapeutic levels of AAT in the mouse. That translates to maybe lifelong expression in humans, and actually that's really quite important when we talk about neonates. It's safe, okay, guys? It's a very safe, no safety profile. Some people have a horror show, and I've been talking this before, and they worry about lentivirus and oncogenesis and things like that. Well, in the early designs of the lentiviral stuff, indeed, there were sadly cases of oncogenesis. That was due to the delta 3U prime regions being a little bit longer and the pathway which they induced oncogenesis. Ingenuity is one of those vectors which has the main part of that deleted, and the risk of oncogenesis has been reduced. And actually, for all of the drugs that have opportunities of every dose that's been given to a subject since that time, there hasn't been a single evidence of oncogenesis from a third-generation lentiviral vector of the self-inactivation type, as are we. So we're really confident about that. The packaging capacity is great. So look, okay, we've got twice that of AAV. Efficacy is retained. Again, a unique feature of Ingenuity. Some vectors, you get viral-induced immunity, and you can't redose after the first dose. That's actually challenging in therapy. It's also challenging in dose finding to start with. Because if you get immunity from your first dose, how do you know what dose you're going to give? So that actually goes back to development as well. But the Ingenuity vector can be redosed multiple times with no loss of efficacy. There's a technical reason for that, but we might talk about that later. That could be a good question to ask, actually, later on. And speaking as an engineer, again, for de-risking, Kyril's in the room, so we have a great partner in Oxford Biomedica locally who has made the vector in the past. This has already been done at GMP. It is being done at scale, supporting the BI program, and very helpfully, independent of indication, 80% of the vector is the same in every time. So 4 of the 5 plasmids which go up into the makeup are common. That's really helpful for economies of scale. We talked about Boehringer Ingelheim. Here's some posters from the European Society of Cystic Fibrosis earlier this year confirming that it's going to clinical trials, hopefully before Christmas, we can expect. It shows, prior work shows the functionality of the gut organoids, that it worked. This graph on the top left here actually is another feature of the platform, is that we can dial up or dial down the longevity of duration depending on the promoter. We have lots of IP over that. And in the past, the guys did a good deal and they got some good funding. But let's talk about reality and what's our first target. So we have 2 programs which are closely related, and these are neonatal surfactant protein deficiencies. And these proteins are fundamental on the 82 cell surfactant metabolic pathway in terms of how they secrete. Infants present with respiratory distress syndrome in the ER. I have been there when that has happened on a personal capacity, and it's not very nice, I can tell you. Right? It's not very nice to see that. But what happens then, the child, or the parents go through the joy of birth, the child gets whisked away and they get ventilated. They get stabilized and they get ventilated. And then you start to worry a little bit, and then the genetic test comes back, and in reality, there's nothing that can be done. And what happens is people have a think about it. Lung transplants for little people are really rare. Heart transplants, which my daughter benefited from a little bit later in her life, were equally rare, and a lot of things have to line up for this. So unfortunately, once this genetic diagnosis is confirmed, 99 times out of 100 treatment is withdrawn and these children die. That's not a great situation to be in. The 1-year survival rate is 0, but what can we do about it? Well, with Deborah's work and her team's work in Oxford, we already have the solution. People have been saying today, we can, we will, we must, we are actually, as we are, and showing some comparative response to other approaches in the space. On the left-hand side of the graph here, you can see what we've been doing. We've had a brilliant knockout mouse model. We have pre-treated our mice with AVG002, in this case for SPB, and if you look towards the blue line, we get a very good survival rate out to the full year of the mouse. When you compare it to others, or wild type, be that mRNA or DNA or AAV, those potential therapies are not viable. Ours is unique, it is world-leading data, backed up by the histology when we look at the untreated control versus the disease-induced and actually the disease-treated induced. You can see we're effectively, be very careful about using the word cure, because that's not really allowed, but we're certainly having a disease-modifying impact, which will likely mean that these children could survive. So what are we doing about that? Well, commercially, we have now applied for orphan drug designation. We've also applied for a pediatric rare disease designation from the FDA, and should and as we believe we'll become successful on that, we'll get a pediatric rare disease as our priority review voucher, which we can sell at a later date. These features you see here are translatable across other indications. At this point, I think the battery has just died on my pointer, so let's try and do it on here. Excuse me. Okay, I like to build the dashboard, because it's all right saying it's great science and things like this, but actually what does this really mean from a business case? And in a business case, when you're doing any disease, rare disease or others, you need to think about your prevalence, your clinical trial, standard of care, CMC, and then put it all together, in this case, in the commercial box on the left. So this is an ultra-rare disease, these indications, 10 on SPB lethal, 30 to 50 on ABCA3 lethal, but there may be an awareness goes forward with our patient groups and our patient pathway. We could lead to more, but in the round, let's say 50 children per year with both opportunities. The clinical design is straightforward. Actually, the work at the Rosalind Institute, which has been done with Jerry McLachlan and Chris, we've actually been using little piglets, right? They're 3 kilos in size, about the same size as a baby. You can put them in an MRI scanner and you can work out the installation protocol, and that helps you with your dosing and your design. We will pursue a basket trial for this. Why do we need to do a basket trial? Well, we need to go and find these children, and if we find an SPB child or an ABCA3 child, we really need to be able to offer either or treatment. So we're doing that. And usually, this will be an instantaneous readout. So if the child comes off the ventilator, it works. The current standard of care, nothing exists. The CMC is sorted. And when you throw it in together, what am I positioning here for us? We're meant to do 40 products, right, I think, in 10 years. Is that right? Well, here's 2 that can be done in 3. 2 that can be done. So 5% of Harrington Oxford's target is on this slide as we speak. And we'll position these for the preferred and only disease-modifying products for neonates with lethal SPB or ABCA3. It's also important when you're at this stage to think about commercial, but independently. So the team have undertaken what's called a HEMA assessment, Health Economic Market Assessment, with Catapult. I can't give you the details of that today, but what I can say that because of the approach, we're in the highly specialized technology group, and there's a higher willingness to pay threshold in terms of maybe up to £100,000 per quality life-adjusted year. And what that means for us is we could probably put this opportunity in a couple of $100 million base revenue. Full cost of market for us for 2 products is as little as $27 million. That's a 10x in a single year. That's a good bet, if you ask me. Moving on, the last few minutes to talk about alpha-1 antitrypsin. This is both lung and liver disease, but we're very much focused on the lung disease. Why? Because the vast majority of patients who present with this have lung disease. The current maintenance therapies right now are human-derived plasmas. You go for a weekly in vitro injection. They're suboptimal, they're not widely reimbursed, and quite frankly, they don't work very well. Other people are looking to do this by maybe lowering the cost, providing a recombinant version of that protein rather than the human-derived. In reality, a 3-week IV versus a 1-week IV, and the cost of goods not being that different, it's not really making an impact. So it's a growing opportunity, only 10% being diagnosed, but the key thing here is that the AAT lung disease is dominant, and I'm going to be a bit controversial. Many a model that's used in science has confirmation bias in it because it's used to serve a purpose. So if you've got a gene editing or a base editing or a down regulator or an up regulator, and you happen to find a model which is easy to show the effect of your platform, well, it's quite easy to show that as well. Let's do that. Let's see what you can do. So in the case of AAT, serum levels, as opposed to AAT levels in ELF and epithelial lining fluids, they're often used a lot because they're easy to sample and quantify. But there is an emerging view of school of thought that that means nothing with regards to actual lung health. And we are the only company in the development competition race here that are reporting measures and correlations, again, commercially relevant models. It's really important to understand that lung index behaves differently, and the clinicians now are suggesting that actually treating those with liver disease doesn't actually necessarily have an impact on other disease. Some data, I'm running a bit short of time. We have, in this case, there isn't a decent mouse model, so we built our own. Bottom left-hand graph, again, 2 years expression in a mouse. The middle graph is actually quite important. Here what we've done, we've compared against the most advanced commercially relevant candidate, alvelestat from Mereo, which is an oral human neutrophil elastase inhibitor. We sought to do that to get commercial relevance, and also because the FDA have told Mereo that if that was successful, how they would get it to market. The problem with Mereo compound, it has dose-related side effects, so it will not go forward fast-paced too. So the untreated animal on the first column, H&E only. So what we've done here, we've had our model, we've then insulted the animal, and we've caused bleeding in the lungs, and then we've started to measure hemoglobin. You can measure that, and you get your dose responses on the right. In the untreated model, that's the thing we have to beat. You see on the 5 or the 10 in the blue circles, that's Mereo. The red circles represent AVG001, which we treated the animals with 2 weeks prior to the insult challenge. And as you can see, with a 1 ENI dose, we are better than the leading commercial standard at this particular moment in time. So great, it's really good. Even more impressive, depending on how you view the world, it says 28 days expression in NHP. That doesn't sound very good next to actually the 2-year in the mouse, but actually it's exceptional. This was a non-immunosuppressed model. Our academics actually were flabbergasted that we got past day 7 And actually, this is a cracking result to still maintain therapeutic levels of AAT in the lung in an NHP. There is some competition, as I've said. I'll just put up that to be, in all fairness, there is a bit of a race going on here. Some are further ahead than us, some are further or not. Be that RNA, be it CRISPR, some small molecule approaches, alternative vectors. The snapshot on there is here in that when you're looking at a preferred target product profile, convenience, measuring things in the right place, very infrequent, maybe once a dose with 2 years shown in thing in a mouse. The efficacy in the real thing that matters, which is in lung disease, not in serum levels, and a great safety record too, I think will be very good. My dashboard for this is quite simple as well. This is a large opportunity. These are hundreds of thousands of patients. It's growing. A clinical trial there. CMC, again, can be leveraged. The one thing I'll pull out on the right-hand side here is, apart from the positioning that directly targeting the lung, there's a big upside in COPD coming. 2 more slides. Right, I do. 2 more slides. Additional COPD, 10% of patients with COPD actually have AATD in the lung, not the liver. So why fix the liver if the liver isn't broke? Last 2 slides is simple. This is my team. Theodore inspired me with Tolkien yesterday. This is my team of superheroes. And in Tolkien's there's no prizes who might win the role of Gandalf on this particular slide. I'm sure Eric would say. There's a bit of competition for Arwen, Deborah and Uta, and the rest of us are just hobbits holding it all together. All right. And my final slide is actually a repeat of the first. Focus on the detail here is that the most important thing for us here is we're trying to do something. We can do it today. We are the only lung-targeted inhaled gene therapy company, which I think is going to work. It fits everything. Thank you to Harrington. Our programs are de-risked, and we look forward to your help in raising the extra £50 million that we need to go to the next stage, Diana. Can I leave you with that thought?
In partnership with our colleagues at Oxford Science Enterprise.
Of course. Thank you.